Hello, everyone, and welcome to the Next Generation ODT presented by Catalent. I'm Michael Gibney, editor of Fierce Markets Life Sciences Group, and I'll be moderating this webinar. Our speakers today are Leon Grother, Principal Scientist at Catalent Pharma Solutions, and Rosie McLaughlin, Director of Scientific Affairs at Zydis. You can read their full bios on the right side of your window. Just a few technical notes before we begin. If you have trouble reading a slide, please hold and drag the right corner of the slide window to enlarge. Please also disable your pop-up blocker to participate in the interactive parts of this presentation. If you'd like to download the slide deck, please click the Download Slides widget at the bottom of your screen. This webinar is being recorded and will be available on demand within 24 hours after the event. We will follow the presentations by a Q&A session. Please submit your questions during or after the presentations using the Q&A box at the right of your window. Okay, now let's begin. Leon, please go ahead.
Okay. Hello and good morning, good afternoon, or good evening, wherever you may be. Firstly, I'd like to thank you all for taking time out to view this webinar. The aim of this webinar is to provide an update on Zydis technology and advances in its capability. Firstly, I'm going to discuss some background regarding the Zydis freeze-dried orally disintegrating tablet, including its advantages, and a brief overview of the manufacturing process. I will follow this with some discussion of recent advances in the technology which will allow us to expand the range of APIs and excipients we are able to work with. I will then hand over to Rosie McLaughlin, who will discuss the Zydis Bio and the next generation of ODT, Zydis Ultra. So what are the advantages of ODT technology, and why does the improved speed of the Zydis ODT matter? There's the pediatric challenge. I have an 8-year-old daughter and a 14-year-old son who is prone to sporting injuries. Getting either of them to take standard tablets or capsules is difficult, and I don't believe my children are unique in this respect. Getting them to take suspension or melt-in-the-mouth tablets is much easier for them and for me. Geriatric patients can often have difficulty swallowing, and patients with Parkinson's disease are particularly afflicted with the difficulty swallowing. The use of ODTs can help with the delivery of medication to these patients. For the mental health challenge, it is important for the caregiver to be sure that the patient has swallowed the medication. The use of ODTs is advantageous as it is not possible to hide this type of dosage form in the mouth and take it out later. This is known as cheeking and can be done to simply avoid the medication or can be done to collect tablets for various reasons, including suicide attempt. I find the animal health challenge is even more difficult than the pediatric challenge. One can try to reason with children to some extent, but you can't do that with animals. I have cats that I have to give tablets to from time to time, and this is usually an ordeal. I have to wrap the cat in a towel or blanket, place the tablet in its mouth, and hold its mouth shut for about 30 seconds. Sometimes after doing this, and still getting my hands covered in scratch marks, the tablets still drop out from its mouth intact. With a rapidly disintegrating ODT, the tablet will disperse as soon as it is inserted in the cat's mouth, thus preventing it from being ejected. I'd like you to try and envisage yourself as a patient or caregiver in these situations and consider what difference the speed of disintegration time would make. In each scenario, surely faster would be better. This slide shows that as well as the specific groups I previously mentioned, the geriatrics and children, etc., there is also a significant proportion of the general population that have difficulty swallowing. Compliance on the move may also be enhanced. When traveling, access to water may not always be readily available, and in these situations, ODTs would be a more convenient dosage form as they are easy to take without water. Similarly, indications where rapid onset of action is required may also favor the use of ODTs. Taking medication for insomnia is again suited to avoiding the requirement for water, as are other indications such as migraine and other nausea-causing illnesses. The further information supporting these comments can be found in the reference at the bottom of this slide. In this slide, we've provided a further example of better compliance for the Zydis ODT versus the standard tablet. Zydis Zelapar was introduced into an already genericized market. The differentiated Zydis dosage form established a leading brand position, and as this data shows, there was a 22% improvement in compliance for the Zydis ODT compared with the existing dosage form. So I've talked through some of the advantages of the ODT technology, and I'm now going to talk in some depth on what Zydis ODT is. It is the world's #1 orally disintegrating tablet. It is typically disperses in less than 3 seconds on the tongue. It is a proven technology with product marketed for over 30 years. There are over 35 products launched in over 60 countries. Over 1 billion doses are manufactured annually from our UK Swindon facility. I'll talk in more detail on taste masking and pregastric absorption in subsequent slides. In one of the earlier slides, I asked you to envisage the difference in mouthfeel and convenience of an ODT that disperses in 3 seconds versus one that disperses in closer to 30 seconds. This slide again highlights that the increased dispersion rate of a unique freeze-dried Zydis ODT can lead to faster onset of action and may enable pregastric absorption for some suitable APIs. The data in the table shows the Zydis dosage form has a very rapid disintegration rate, whereas the competitors tested here are much slower with disintegration times in excess of 15 seconds. The competitor mouthfeel is also described as chalky or gritty, whereas the Zydis formulation mouthfeel is described as smooth. The Zydis fast-dissolve technologies are categorized into 3 groups. The Zydis ODT, which I will discuss on the following slides. Rosie McLaughlin will then discuss Zydis Ultra. That allows greater doses to be improved and improved taste masking. Rosie will also discuss Zydis Bio, that delivers large molecular formulations for delivery of allergens, peptides, and viral vaccines. This slide lists aspects of some of the products currently marketed in the Zydis format. As you can see, they are predominantly fairly low doses of small molecule APIs, though a product with a 200 milligram dose strength has been previously commercialized. Note also, the commercial product also includes a peptide for the treatment of bedwetting and protein extracts for the treatment of various allergies. If you look at the indications listed on the table, you will see that there are these are wide-ranging, although the Zydis product is particularly well suited to many of the indications. For instance, nausea and motion sickness are helped by not requiring water when taking the medication. Schizophrenia is helped as it is easy for the caregiver to prevent taking the medication, and for Parkinson's, there is the ease of swallowing that is of much benefit to these patients. Now moving on to how the unit is manufactured. Here we have the basic Zydis formulation. The matrix former typically contains gelatin and mannitol, which give the unit its strength and elegant appearance. The gelatin may be from a bovine or fish source, and we are also able to use non-gelatin polymers. Other formulation excipients may also be included. These include sweeteners and flavors, as well as other excipients that may be found in pharma formulations, such as colorants or pH modifiers. The photo at the bottom of this slide shows electron micrographs that highlight the high porosity of the formulation. It is the combination of the excipients used and the high porosity that allow the formulation to have very rapid dispersion times. On this slide, we have a simple schematic illustrating the Zydis manufacturing process. The Zydis excipients, such as those mentioned on the previous slide, are added to purified water and mixed. The API is added, and depending on its solubility and the product dose, either a solution or suspension is produced. This mix is then accurately dosed into preformed blister trays using precision positive displacement pumps. The blister trays are then passed through a liquid nitrogen freeze channel, where the mix within the blister pockets is rapidly frozen over just a few minutes. Following the rapid freezing, the blister trays are stored in a freezer prior to loading into a freeze dryer. Under very low pressure, the drier shelves are heated so that the ice crystals transform directly from the solid state to a vapor state. By avoiding the liquid state, the product retains a highly porous structure. When the dried product is placed in the mouth, the saliva is able to rapidly penetrate this porous structure and rapidly dissolve the structure-forming ingredients due to the high surface area available. Okay, on this slide, we have a more visual representation of the manufacturing process from blister-forming fruit sealing product. As well as traditional chemistry testing, such as assay, dissolution, moisture, etc., there are a number of product characterization techniques that are routinely used to develop a robust Zydis product. We use X-ray diffraction to determine the crystalline state of the API and some of the excipients. We use differential scanning colorimetry to determine product melting point and other thermal events that help us to characterize the freeze-drying cycle development. Dynamic vapor sorption is used to determine what humidity the product can be exposed to. This is important as freeze-dried formulations can be sensitive to high humidity environments where they can shrink and lose their rapid disintegration characteristics. Appropriate formulation and packaging choices allow Zydis product to be marketed in all geographic regions. I mentioned earlier that Zydis can, in some cases, enable faster onset of action and reduced side effects via pre-gastric absorption. The top two plots on this slide show how sledgeline can be delivered via Zydis ODT to avoid first-pass metabolism and therefore have lower undesirable metabolites and reduced side effects. In the first plot, it can be seen that a 1.25 milligram dose delivered via Zydis has a similar area under the curve as a 10 milligram conventional tablet. In the plot showing the metabolites, it can be seen that there is a tenfold reduction when the dose is delivered via Zydis. As the metabolites contain amphetamine, the Zydis product is safer and more convenient to take as the associated side effects are significantly reduced. It is important to point out that not all drugs can be formulated to achieve pre-gastric absorption. This is dependent on the API dose, solubility, lipophilicity, and molecular weight. If the API is suited for pre-gastric absorption, then the formulation can be modified to further enable this. The bottom plot is an example showing bioequivalence between an API delivered via a capsule formulation and the Zydis formulation given with and without water. As you can see in this case, all three options are bioequivalent. This slide shows a recent advancement in the Zydis technology that allows two incompatible ingredients to be delivered in the same tablet. Two solutions or suspensions can be dosed into the same blister pocket and frozen before they have time to interact. This allows incompatible APIs to be delivered in combination product, or could allow an API to be delivered with an incompatible excipient. In this example, a blue dye was added to one layer to help illustrate the separation between the two layers. Okay, here we have another recent development where we have demonstrated that we can formulate relatively high doses of oils as emulsion formulations that can then be freeze-dried to provide an elegant Zydis unit that retains rapid disintegration properties. In this example, a Zydis formulation was prepared containing 15% olive oil. This was dosed and freeze-dried to produce elegant Zydis ODTs containing 75 milligrams of the oil, and this formulation retained its fast disintegrating characteristics. Such formulation strategy could be used to deliver an oil-based API, or could be used to deliver a poorly aqueous soluble API dissolved in a compatible oil. One of the challenges of formulating a successful ODT is that of taste masking. Many drugs have an unpleasant taste or sensation, and as ODTs release the API in the oral cavity, steps often need to be taken to make the product palatable. There are several taste masking strategies that can be utilized, and the chosen strategy depends on the extent of the unpleasant taste and also on the dose and characteristics of the API. Where the API taste is only mild or moderate, the use of an appropriate flavor and sweetener is often sufficient to provide an acceptable tasting product. Where the taste is very bitter or there is a sensation such as numbing or burning, more advanced taste masking techniques need to be employed. The formulation pH can be manipulated in order to reduce the solubility of the API, though this is only suitable for some drugs that are ionizable. The use of ion exchange resins has also been successfully demonstrated in Zydis formulations. Under appropriate formulation conditions, the API forms a complex with the ion exchange resin that is insoluble at oral pH, but releases the API rapidly under acidic conditions in the stomach. Ion exchange resin for taste masking is again only suitable for ionizable drugs. Another advanced formulation technique that we have recently demonstrated with Zydis ODT is use of cyclodextrin. These have a cylindrical molecular structure into which some API will preferentially associate. The API within the cyclodextrin cavity does not come into contact with the taste receptors on the tongue and is therefore not tasted. Here we have a case study where we have taste masked using cyclodextrin. This slide shows results of a consumer study and a probe bioequivalent study performed on a Zydis formulation containing cetirizine, which is known to be a very bitter tasting API. This formulation contains cyclodextrin, which was found by the taste panel to provide very good taste masking. It is important that the taste masking techniques used do not have a negative impact on the release of the drug and its pharmacokinetic profile. The area under the curve has demonstrated bioequivalence between the taste masked cetirizine Zydis formulation and the standard cetirizine tablet. I will now hand over to my colleague Rosie McLaughlin for the remainder of this presentation to discuss Zydis Ultra and Zydis Bio.
Okay, thank you, Leon. I will now introduce a new exciting development for Zydis, referred to as Zydis Ultra. With Zydis Ultra, we're now able to incorporate the coated API into the Zydis platform for enhanced taste masking and at higher dose strengths. Previously, with Zydis, it wasn't easy to include coated APIs that have been coated by conventional means, such as fluid bed, because of the swellable polymers that tend to be used in that process, which, when included in the Zydis formulation, would leach drug during the aqueous stage of Zydis processing. However, it's now possible to include the coated API by the use of a proprietary novel dry mixing process, which we use to coat the APIs. In this dissolution release profile here, we are comparing Zydis Ultra made, the two bottom curves, made using coated API by the novel coating process compared to API coated by conventional means. And you can see here that initially, with the new coating process, the dissolution release profile is retarded, which is indicative of taste masking. But at the same time, we're still able to achieve the desired immediate release profile by meeting the USP criteria. So, how does the novel dry coating process work? We charge the coating polymer particles, the host API particles and milling media into an acoustic mixer, and the acoustic mixer vibrates and accelerates the particles within the mixing vessel by as much as 100 times G, so 100 times the force of gravity, so it's a significant acceleration. And initially, the polymer particles form as discrete deposits onto the host API particle, and by the action of the vibrations of the mixer and the impact from collisions with milling media, those coated particles are then deformed into a continuous polymer layer, providing the taste masking. There's no solvent involved, and the most significant aspects of this process is that the coated API potency is in the region of 70 to 85% weight, which when compared to conventional coating, that's normally in the region of about anywhere from about 40 up to occasional 80% weight. So by having a coated particle with high API potency, that also allows us to achieve the higher dose strengths within the Zydis format. And secondly, of particular significance for an ODT formulation is the ability of this process to coat small particles, roughly around 100 micron. Again, compared to conventional coating techniques, the particle size of the resulting material is normally anywhere in the region of about 200 to 400 microns, which would feel extremely gritty and coarse if included into a coated into a ODT formulation. Using this technique, we've been successful in the taste masking of an ibuprofen 200 mg formulation and also an APAP or paracetamol, as it's known, 325 mg formulation. They're extremely well taste masked. There's a picture of a 50 mg Zydis units containing coated ibuprofen to the left, and an SEM photomicrograph of the coated ibuprofen particles themselves. I think you'll just about discern from that image how well each individual particle is coated. So hopefully that's given you a good overview or an introduction to Zydis Ultra. I'm now going to move on to the potential for using Zydis for the sublingual delivery of proteins, peptides, vaccines and allergens. The main advantage of the sublingual route over an oral route for these types of molecules is that it avoids the harsh environment of the GIT. So in your gut, you've got the acidic pH, you've got the peptidases and other proteolytic enzymes. And you've also, by delivering, if you deliver to the gut, you actually get quite a significant dilution of the dose that you've applied as well. So if it survives the pH and proteolysis, it's also going to get diluted. So sublingual administration completely avoids the vast majority of those effects. The other advantage of sublingual administration is our ability to use bioadhesives, so we keep the dose in the locality of the administration, and also the ability to use absorption enhancers to improve the oral uptake. In this next slide, we've just got I thought I was on to the next slide. In this slide, it describes some of the advantages of formulating proteins and peptides into the Zydis format. Obviously, as a lyophilized process, we're able to use low processing temperatures, which is very gentle towards the protein and peptide being incorporated. Again, the Zydis dose format has very low water activity and avoids hydrolysis of the protein. And together with the use of an all-aluminium packaging blister pack, we can achieve long-term room temperature storage for several protein and peptide compounds. So now moving on to an example peptide, which is salmon calcitonin, widely studied in the literature. Here you can see on the right a comparison of the pharmacokinetic profile of a Zydis formulation delivered sublingually compared to the same formulation given by Gavard. So when we give by Gavard, we make sure that the tablet goes into the gut, and that you can see the PK profile in the dark blue there when administered directly into the gut. When administered sublingually, we see a significant increase in the bioavailability. And following on from our success with proteins and peptides, we thought to ourselves, well, why not try to use this for oral delivery of vaccine antigens, which the vast majority of our peptides in nature, protein peptides in nature. So, but why do we need an oral vaccine in the first place when injectable formats of vaccines have been highly successful for decades? Well, there's some very good reasons and advantages that oral vaccines may provide. The first one is obviously compliance. Everybody would much prefer to take a tablet than be injected. And also there's been some poor press around vaccines and its links or reported alleged links to autism. And that has led to outbreaks of poor compliance, certainly in the UK and US, and significant measles outbreaks. Potential for an improved safety profile versus injectables, especially in the developing world, where maybe syringes and hygiene is not so controllable as it is in the developed nations, and avoiding needle stick injuries also. But a significant driver for oral vaccines is that most of the diseases that we're exposed to actually initiate at mucosal surfaces. And if we can induce a mucosal immune response as well as a humoral antibody response, then it is believed that this could be certainly more effective against mucosal infections such as flu, pneumonia, and HPV. And the last 2 points there, obviously with the Zydis or an oral presentation of the vaccine, the speed of distribution, the ability to avoid cold chain distribution, much more conducive to massive immunization programs, with the potential that less qualified healthcare professionals can administer the dose. So this is some work that we've done internally using flu as a model vaccine antigen. And in the graph on the right, you can see that following sublingual immunization using Zydis of the flu at days 0, 10, and 20, the mice were challenged intranasally with flu. And following that period, you can see that the body weight losses in all of the Zydis formulations, but particularly in the top one, is much less compared to the unvaccinated and infected control, which is the bottom line on the graph. And body weight is an indicator of the extent of the disease, or body weight loss is an indicator of the extent of disease. And you can see that the top Zydis formulation there was hardly any change in body weight at all in that period. And supporting that is the graph on the left, which for one of the Zydis formulation, we were able to demonstrate 100% protection, i.e. 100% survival of all the animals in that study. So we believe this data shows significant promise and potential for Zydis in the vaccine arena. So I think both Leon and I would like to thank you for your time listening to the presentation today. I hope you found the presentation interesting and informative, and I hope we've managed to relay the extreme flexibility and diversity of the capability of the Zydis fast dissolved dose form, from enhancing bioavailability of small molecules to the delivery of proteins and peptides on vaccines, and the significant advances we've made using Zydis ultrafructose masking. So thank you very much, and we would now like to be able to answer any questions that you may have.
Great. Thank you, Rosie, and thank you, Leon. So let's move on to the Q&A. There's still time to submit your questions using the Q&A box at the right side of your window. We have lots of great questions already, and we'll try to get to as many as possible. First question: what is maximum amount of small molecule possible to perform with Zydis?
There isn't a black and white answer to this. As I mentioned earlier in the presentation, we have commercialized a 200 milligram dose, and we've also developed a 300 milligram dose to late stage on scaled up equipment. What we would tend to find is that the higher doses will produce a larger tablet, and the dispersion characteristics in the mouth will become less pleasant as the tablet size and dose increases. So it is somewhat API dependent as well, in that the higher doses we can produce tend to be for suspension formulations rather than solution formulations. So with a API which is highly soluble, the limit is probably closer to 100 milligram, and I'd say it's probably about 300 milligram for a suspension formulation. But, if a customer came to us with a 320 milligram, then, we would do a feasibility study to assess the impact of that size on the dispersion characteristics and cost of the product.
Great, thank you, Leon. Next question: will Zydis emulsion formulations improve the bioavailability of poorly soluble APIs?
We haven't generated any data as yet to prove this. However, this is quite similar to technology that's been used with soft gelatin formulations for many years, which is well proven, where they've included very insoluble APIs dissolved in an oil or a surfactant type emulsion and put that into a soft gelatin capsule. I don't really see any reason why this would be any different for a Zydis formulation.
Great, thanks. Next question: what would be an average shelf life for an oral vaccine?
The average shelf life we would be hoping to achieve, well, if I can answer that in another way, for allergen antigens and for proteins and peptides, we have already demonstrated up to about 3 years room temperature stability. So when you were talking about vaccines, it depends on what type of vaccine we're incorporating. If it's a vaccine antigen, such as a recombinant protein or a split vaccine, then we should be able to achieve similar shelf lives to proteins and peptides. If it's a live attenuated vaccine, then we have less limited experience with that. But, we should be targeting at least around 12 to 24 months stability. But as I say, we have less experience in that arena. Our focus has been on split vaccine antigens.
Thank you, Rosie. Next question: is it possible to apply Zydis Bio to to insulin for diabetic patients?
Potentially it is. We would obviously need to do the feasibility around that. I mean, I know already there have been some efforts in that area, and I think it would be a case or an interesting candidate for a feasibility study using the Zydis platform.
Great, thank you. I'd like to remind everyone there's still time to submit your questions using the Q&A box at the right side of your window. Next question. Is the novel dry coating process applicable to all APIs, or are there any technical limitations?
The dry coating process is applicable to a wide range of APIs, including soluble APIs, which are particularly problematic during conventional coating. In conventional coating, if you've got a soluble API, then you have to make a careful selection of which solvents you're using to apply the coating polymer. With this novel dry coating process, that's not a concern. So potentially, you know, we can coat a wider range of APIs compared to conventionally coated API by fluid bed.
Great, thank you. How many commercial products are there for Zydis Bio?
Well, I think that was shown on one of Leon's slides, where we certainly have at least 2 on the market, and there are several in the pipeline.
Yeah, there's one peptide API which has been on the market for a number of years now, and there's a number of different allergen formulations which are on the market, which again come under the Zydis Bio umbrella.
Thank you. So what range of vaccines will be applicable for inclusion in Zydis Bio?
We believe a wide range of vaccines will be applicable. Our main experience has been with whole inactivated vaccines and with split vaccines, and we believe it would be also applicable to recombinant protein vaccine antigens and virus-like particles and virosomes. So it actually has potential applicability across quite a wide spectrum of vaccine types.
Thank you. Pardon me if I mess up the pronunciation of this word, but what is the friability of the tablet? Do we need special packaging?
It's actually surprisingly not friable. Friability is when a tablet sheds tiny little particles from its surface. In that sense, Zydis is not friable at all. It is obviously of a softer consistency than a conventional tablet, but it's sufficient to be taken out the pack, handled, withstands transport. But yes, certainly the packaging helps in that respect, but in terms of friability, it has low friability. It doesn't chip or shard like a conventional tablet would.
Thank you, Rosie. What is the maximum dosage form you are able to commercialize with Zydis Ultra?
At the moment, we have demonstrated dose strengths up to 325 mg, as you saw in the presentation. We've also done some work with the 400 mg presentation. We believe we may be able to increase that to 500 mg, but as Leon previously explained, it then becomes a question of the acceptability of the size of the tablet.
Great. Next question. Is Zydis appropriate also for ODT with modified release of the API?
In principle, yes, it is. Our focus at the moment is using functional coating for taste masking, but there is potential there.
Great, thank you. What are the typical timelines to develop Zydis ODT for FTIH study?
Could you repeat that last piece, please?
Sorry, yes. For FTIH study.
Is that first time in human? I don't think we understand the question in terms of FTIH.
Oh, I'm sorry, yes. I'll repeat. So what are the typical timelines to develop Zydis ODT for the FTIH study?
Yeah, it will be first time in human.
Normally we would do a feasibility study, which would take approximately 3 months to get to a suitably robust formulation to then taken into a clinical study. And following that, we would probably need another 3 months to prepare all the onboarding and GMP requirements. So probably approximately 6 months in total. But that would be for bench scale studies as opposed to taking the formulation through scale up and validation, etc. So that would be a first-in-man phase 1 type study, which we would hope to be able to do within 6 months from initiation of the study.
Okay, thank you. Next question. How do you prevent the peptides and proteins delivered by the sublingual route from being swallowed?
Yeah, we do that by the fact that Zydis, the major matrix former, is gelatin, which is quite mucoadhesive in itself. And we can also augment that with other bioadhesives such as ethyl cellulose. So that actually retains the protein and peptide in quite good contact with the mucosa and inhibits salivary wash in terms and prevents any significant swallowing of the protein and peptide, or indeed small molecule if it's delivered sublingually as well.
Great, thanks. Have you commercialized any products using Zydis Ultra technology?
Not to date. It is in development, and there are molecules in the pipeline, but to date, no, we haven't commercialized Zydis Ultra.
Okay, thanks. Now, this next question is a little vague. How do you get your samples?
I'm assuming this is, have we got samples, the marketing samples we can send to people? And if that is the question, then yes, we have a large stock of samples that we can send out. So people are welcome to visit the Catalent website and request these, or contact myself or Rosie by the details provided.
Okay, thank you. Where are your Zydis facilities located?
The main facility is located in the UK, in Swindon, but we also have capability in Somerset, New Jersey, but the vast majority of commercial products and the main capability resides within the Swindon, UK facility.
Okay, thanks. Can you describe the particle size of the taste masked particles?
Yeah, because the dry coating process allows us to coat smaller particles in the region of about 100 micron, then the final coated particles will be around 100 to 150 microns in size. And this is related to the fact that the coating polymer layer is relatively thin, and that's one of the reasons why we can achieve such high potency with the final coated API.
Thank you, Rosie. Next question, what is the percent uptake of a peptide to kDa with ODT?
Yeah, that is a difficult one to answer because each molecule as a peptide, would have different phys-chem which would need to be taken into account in terms of its bioavailability. So it would have to be determined on a case-by-case basis, but I would refer you to the salmon calcitonin data, which is about 3 to 4 kilodaltons, where we did see, an appreciable uptake and bioavailability in that format. So I would think a 2 kilodalton peptide would exhibit some appreciable bioavailability, but it's hard to quantify that without actually doing the studies on that particular peptide.
Great, thank you. What is the ideal particle size distribution required for dry coating process?
Yeah, well, obviously from a mouthfeel perspective, we would like to keep the particles below 150 microns. So the critical criteria for us is that upper particle size fraction. That's where we target, is to keep the particle size less than 150 microns.
Next question, could Catalent provide support to calculate the amount of drug by sublingual route versus oral route?
Yes, through third parties, we could do that. We don't have the facilities on site to do that, but from prior studies, whether they be ex vivo techniques or in vitro animal studies, we have the necessary network to enable that to happen.
Great, thank you. How acceptable is Zydis for pediatric dosing? Have any clinical studies been conducted in infants?
I'm not aware of any specific studies in infants. Not sure if Rosie is. However, we consider the Zydis ODT to be ideally suited for pediatric use, and probably more suited than other ODTs for younger children, simply because the ODT disintegrates almost instantly when it's placed in the mouth. There is much less of a choking hazard than there would be with standard tablet or an ODT, which takes 15 to 30 seconds to disperse. So as the Zydis ODT is placed in the mouth, it's virtually akin to having a suspension formulation placed in your mouth in terms of the dispersion rate.
Thank you, Leon. Next question. Are there any specific storage requirements for a Zydis-based product?
No, not really. In the all-aluminium blister format, it's very well protected from any moisture ingress, and so most of the products don't have any special criteria for storage other than room temperature, normal ambient room temperature storage.
Great, thank you all. That was our final question. We had a lot of great questions today, but we'll be getting back to everyone who submitted personally after the webinar. Thank you for attending this Fierce Live webinar and submitting so many great questions. I'd like to thank our speakers for participating and Catalent for presenting today's webinar. This webinar has been recorded. You will be able to access the recording within 24 hours on the same page you used to register for this event. Thank you again for joining, and we look forward to seeing you at future events.